Hello everyone and welcome to this video about AP Bio Unit 1. It's basically a review for what the College Board expects for you to know for Unit 1 of AP Biology. And it also has a little bit of an introduction to statistics, which I will cover in another video alongside animal behavior, which will also be covered in another video, simply because I want to really just be getting down to the real meat of the chapter or the unit, so that you have the biggest takeaway from this video. And don't worry, there will be stuff on animal behavior and stats later on in the year as well, but those will be in different videos. Additionally, I'd also like to preface this video by saying that, look, most of the stuff I'm going to be telling you isn't only merely answers. If you came here for that, you will definitely get them, but I'm also going to kind of be explaining why those answers make sense, because on the AP Bio test, which I took last year, the 2022 version of the test, they don't only ask you whether you know the content, but why that content makes sense. So if you watch this video, hopefully I make some of that stuff clear to you, if not all of it. And I'll basically be making these videos for the next 8 units and any subsequent, small chapters within each unit. So, with that being said, let me get to question 1. What are life's organizational levels? You have a biosphere and ecosystem provided to you, and for right now, I'm going to be basically typing out the rest of the answers. You have a community, you have a population, then you have organisms. After organisms, you have an organ system. After organ systems, you have the individual organ, tissues, the cell, which is probably the biggest takeaway from this for this unit and the next unit. And after the cell, you have organelles, which is another big takeaway for Unit 2, but I'll touch on it a little bit here. And lastly, you have kind of like the chemicals that make all those things up. I call them macromolecules, and so does the AP test. Spelled that wrong. Should be okay. Anyways, let's get to every portion of this. What is the biosphere? So the biosphere, to put it in the simplest terms, is Earth. That is what the biosphere is. It is a very large area in which scientists look at, and they don't only look at just one aspect of it. They kind of look at how everything within Earth operates, right? So abiotic and biotic factors. Those are two words you should know. Abiotic and biotic. They're going to be helpful for the rest of the year, but if you're introduced to them now, they're going to be helpful. So abiotic factors, what are they? These are basically non-living things. Weather, temperature, rocks, stuff like that, and how they interact within the area that they're in. And then you have biotic factors, think of just bio, and those are things where you just are looking at how they work together and they're all living. So how does a fish work with a frog? How do those interact in this, and how do those things interact with, maybe the weather? That's a hypothetical. I don't know the answer to that. Don't expect to find it in the video. Next up, you have something more specific called an ecosystem. And before I go further, I'd like to note that these are demonstrations of things called emergent properties. It's not really covered in the AP Bio test, but as you keep on going down and down and down, you're going to be noticing new emerging properties approaching or coming out that each of these specific things have that the previous one doesn't have. An ecosystem, for example, is very similar to a biosphere. You still look at abiotic and biotic factors, but you only look at it in a smaller area. So instead of the world, I'd be looking at it maybe in a forest, right? That could be an ecosystem. Community, almost the same thing as an ecosystem, but now you only look at biotic factors. So if I'm looking at a forest instead of an ecosystem that's a forest, I'd be looking at a forest of just living things, right? So yes, there could be not living things within that forest, but I wouldn't care about what they really do. I'd be only concerned with how the animals within that forest work, or those living things. Then you go to a population. Population is much, much more specific to basically a certain group of living things within there. Like, they can only breed with themselves, kind of. Like, a population would be a population of humans. Humans can't breed with dogs, animals, so on and so forth. They're very, very specific, okay? And also, you'll notice that now we've drifted away totally from abiotic factors. We're only looking at how, humans interact with humans in a population, or how animal A interacts with animal A within the same region. and the region could be large, small, so on and so forth. When we go to organism, that's also a very broad term, but it's just the individual of the population. So if I have a population of humans, I'd be only looking at, one person. So Alex, blah, 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 blah. I'd be looking at the characteristics he has and how he works and how he interacts with society instead of how he interacts with another person as well. That'd be more of a population um, situation. Then you have organ systems. Organ systems. Now, not every organism has an organ, right? Cells don't technically have an organ, but for many living things, animals, humans, so on and so forth, we have organ systems. And what scientists really try to do when they look at that organization level is, hey, look, how does the kidney work with the heart, so on and so forth. You have the organ. What is that organ individually do and contribute to the population or the organism's ability to live? Tissues. These are fine layers of cells basically connected to one another. How do they all work? How do the cells kind of stick to each other? So on and so forth. A cell. Cell is the biggest thing you should remember out of all this for this unit and the next unit. A cell is the smallest unit of life. Another way that I can kind of make an analogy is like the atom is the smallest form of matter that most chemists like to say. So think of a cell as an analogy to that. This cell is the smallest thing. You'd be basically looking at how the cell operates. Organelles are basically the organs of the cell. I'll get more into that in Unit 2's video. Then chemicals that make all those things up, macromolecules. These are just the molecules that construct the cell. Really, this is like the chemistry of the stuff, and this is what the rest of the unit is going to be about. Question number 2. What is the main difference between a prokaryotic and eukaryotic cell? I'm going to draw this out right here, make it very plain and simple for you. There are two really distinct differences. I'm going to call it prokaryotic here, and I'm going to call it eukaryotic here. Eukaryotic. If I spell things incorrectly, my apologies. Prokaryotic, okay, this is the biggest difference, and later on in the year you will learn about more differences, but for the purposes of this unit, you're just going to have to know that these guys are non-membrane bound, or they don't have, yeah, I'm going to just say that they do not have membrane bound organelles. What does that mean? If you can imagine in a human analogy or anatomy, imagine a membrane as this surrounding thing around something. So for example, if I have a heart, imagine I have something surrounding my heart, like a little protector, a shell, okay? Prokaryotics do not have membranes. Eukaryotics, they do have membranes, so they do have membrane-bound organelles. The best analogy that I can give that many students could understand right now for membrane-bound organelles for cells is think of a nucleus. A nucleus is where all that DNA is stored, right? Well, a prokaryotic cell, right, is not going to have that. They're not going to have, and let me draw the wider cell here. This is the nucleus right here. It's that shell, and all the DNA is compact within it. That shell is the membrane-bound organelle. In a prokaryotic cell, the DNA is actually just floating around nice and smoothly. There is nothing protecting it, okay? Yes, they do have a membrane around the actual cell in and of itself, but organelles, remember, are not the same thing as a cell. They're much more compact, and within the cell, they go deeper. So, yes, they don't have membrane-bound organelles. They tend to be smaller and unicellular. So, for example, you're going to see bacteria as a prokaryotic that's typically unicellular, or archaea. Not going to really have to worry about archaea in this test or even in this class, but bacteria definitely. Know that these guys are prokaryotic, unicellular. They don't have membrane-bound organelles. Eukaryotics, on the other hand, multicellular. Think about humans, right? We are composed of multicellular. Well, we are a multicellular organism, right? So they're multicellular organisms. They're way bigger. Think about it. They have to be a little bit bigger to cater towards the many more needs we have. I don't want to get too far into this, but we have more chromosomes typically for eukaryotic cells. There's going to be more chromosomes for prokaryotic. There won't be prokaryotics. Like, think of a bacteria. It's pretty simple. Humans, we're big. We're ginormous. It's got to cater towards more DNA. More processes have to be let to keep us alive versus a bacteria who just needs energy. So, you know, there are those big distinctions between everything. But if you have to take away something, eukaryotics have membrane-bound organelles. Prokaryotics do not have membrane-bound organelles. That's basically it for these one and two. What type of cells are humans made of? I already kind of talked about that. We're made of eukaryotic. Plants are eukaryotic. Fungi are eukaryotic. Bacteria, and here I'll write it in this sheet. Eukaryotic. Eukaryotic. We're also got plants that are eukaryotic. They're different types of eukaryotic cells, by the way, but you know, in the gist of it, they have membrane-bound organelles. Bacteria is prokaryotic. Okay, I'm going to skip 3 to 8 right now and go straight to 9. What are these? Which of these provides evidence of the common ancestry of all life? Ubiquitous use of catalysts by living systems. B. Structure of the nucleus. C. Near universality of the genetic code. D. Structure of cilia. E. Structure of chloroplast. So basically this question is saying, how do I know that there is this one organism, this one cell that basically all of the living things that are here right now are, you know, related to? So how am I as a human being even related, like, to a bacteria cell? I know they're so far away, but we both have DNA. There's got to be something that kind of we all originated from. That's what this question is asking, and the answer is going to be C. Why not A? Each living system has its own use of catalysts. They're all different, so you can't. They all vary. That doesn't really show any form of relation. I don't use the same proteins in my body than a dog would need sometimes. The structure of the nucleus. Remember, how can I prove relativity or a relation to two things when eukaryotics, a type of cell that's alive, is different than a prokaryotic cell? Because a eukaryotic cell has a nucleus and a prokaryotic cell doesn't have a nucleus. That doesn't make sense. D. Structure of cilia. Not all cells have cilia. You'll also learn about what that is in Unit 2. It's basically like the tails of many of these cells that helps like things move around. I'll get into that into the next video. Structure of chloroplast. Again, not all cells have chloroplast. I sure as hell don't have chloroplast, so that would make sense if I want to talk about my relation to a plant. Okay? C. Near universality of the genetic code. This is correct because, and listen carefully because you're going to need to know this in future units as well. If I were to get my DNA, hypothetically speaking, and I were to just shove it into, through the use of biotechnology, shove it into a bacteria cell, okay? There are things called ribosomes in every cell, and these ribosomes would be actually able to read the RNA that is created from the DNA, my genetic code, from my DNA in the bacteria. They would be able to read the RNA that my DNA creates and actually create the protein. Will that protein work? No, but the mere fact that a ribosome can read the stuff that is made from my DNA is the important fact that we are all related because, you know, basically we have some form of relation, that all the ribosomes are the same, so they had to be created from some similar structure. All right, so think about it like that. Okay. 10. Which of the following correctly describes any reaction that has reached chemical equilibrium? A. The concentration of the reactants equals the concentration of the products. B. The rate of the forward reaction is equal to the rate of the reverse reaction. C. All the reactants have been converted to the products of the reaction. D. All the products have been converted to the reactants of the reaction. E. Both the forward and reverse reactions have stopped with no net effect on the concentration of the reactants and products. The actual answer is going to be B. I'm just going to highlight the entire thing here for convenience. Let me explain. So, a lot of people get this wrong. They typically think that both of the reverse and the forward reactions have the same values or like the same amount of substance. That's not true. What is true is if I have two beakers here and I want to, you know, connect them with a tube, right? And I want to make a chemical reaction. If I want to make a chemical reaction, okay, and I'm mixing this particle with this small particle, right, the bigger one and the smaller one, the rate at which those two becomes, you know, let's say this star, and the rate at which this star becomes these two elements, right, or these two particles again, is what a chemical equilibrium means. Not if they have the same constant of values, right? Because, and listen carefully here, I could have only five, 5 stars here and here, and I could have a gajillion amount of these different molecules, and they can, be creating or reacting. These guys, the rate at which these guys, so how quickly these guys become them, and how quickly those guys become the stars is what's important. So if one star becomes those two molecules every 5 seconds as well, two of these little dots must become a star. All right, but if you can see, I have a lot of these still, right? And I have a lot of I only have 5 of these, right? Their values aren't the same, but the rate at which they're becoming one another becomes the same. So in a way, I will always have 5 stars, and I will always have whatever amount of particles I have in here. Okay? So I hope that makes sense. A lot of people get that one wrong, so please, please don't get it wrong. I think that's already a quiz question on one of the AP bio tests. So yeah. I'm gonna skip question 11. It's statistics related, but it shouldn't be too hard. 12. A student has two test solutions. She discovers one has a pH of 2.4 and the other has a pH of 6. Which of the two is a stronger acid? How do I know? Okay. So, what is pH? pH, in simplest terms, is the measure, and I'll write it here honestly. pH is the measure of H plus protons within a system, typically watery solution, all right? And it's base 10. It's base 10. So, when I have 2.4 pH, that actually means 10 to the negative 2.4. Okay, because if you want to calculate pH, you have to go to a base 10 system, so this always becomes the exponent of it. Then I have a pH of, what is it? 6. That would become 10 to the negative 6. If I had a pH of 7, which is net neutral, I would have 10 to the negative 7. All right, 10 to the negative 7. What particles per 1 gram or 1 mole of something, okay? So these are just how many molecules are within 1 mole of it. It's really not a lot, but it's still a significant amount to consider acidity. So when I say 10 to the negative 2.4, I mean that is how many, like I said, how many hydrogen atoms I have, or not hydrogen, proton atoms I have. And 10 to the negative 4 is going to be greater than 10 to the negative 6. I mean, if you just, let's imagine that this was just 10 to the negative 2. 10 to the negative 2 is just 0.1 versus 10 to the negative 6, which is 0.000001, or maybe I got that wrong. But the point is that there's a lot more zeros. 1, 2, 3, 4, 5, 6. Yeah, that's good. Anyways, so yeah, that is why something of negative 2.4, or even negative 2, is going to be greater than something of negative 6. There is a greater amount of protons with 10 to the negative 2, a pH of 2, versus a pH of 6. And if you pass the value of 7, if you go higher, so 8, 9, 10, you're going to be having a thing called a base, or a basic solution. If you have anything lower than 7, you're going to be having an acidic solution. And if you have something that is 7, you're going to have a neutral solution. So I think water is pretty close to 7. What is the difference between a saturated and unsaturated fatty acid? Okay, now we're getting into this to these things about macromolecules, and I will really just try to pull up a photo. Saturated versus unsaturated fatty acids. And there's really just one big distinction behind it. I want people to look at both. I'm going to try to screenshot that, open that up, and then screenshot that. Actually, here, I'm going to screenshot this. Unsaturated fatty acid. Okay, cool. Now, if I hold these two things side to side, and I ask an individual to kind of tell me the difference, it becomes glaringly obvious once I point it out. Whoops, my apologies. You see this double bond right here. This double bond right here. And you don't see that there in all of this. You can ignore this part right here, and you could ignore this part right here. They're the same. But this double bond is actually what's really, really important. Why? Because when we say something is saturated, it means that something is surrounding all of it. And in a saturated fat, we're going to see, oh, hydrogen surrounding every single carbon. Every single carbon is saturated. Every single carbon has a hydrogen attached to it. On the top, on the bottom, and then it has another carbon and another carbon attached to it. Everything is bonded. It's fully enclosed. In this case, we don't have it. There are things missing at the tips of both of these carbon acids. Or, sorry, not acids, carbon atoms. So that is where the word unsaturated comes to be. Okay? And why is this important? Well, if we look at the structures of these molecules, and structure has to do a lot with how a molecule functions, we're going to see a significant difference. This is called, or this double bond right here, these two lines, they're double bonds, creates a kink within the unsaturated fat, making it a little bit longer in a sense. Whereas a saturated fat, because everything is combined together, or it's fully like enclosed, it can kind of be compressed a little bit easier. While this one, you can see the kink, it's already kind of pushing the thing outwards like that. You know, there's this kink right here, it goes longer than, you know, just a line would. Think of that as a line, and think of this as the kink, which impacts this. In Unit 2, you're going to really see why an unsaturated fatty acid makes a huge difference and how that length really impacts stuff. But yeah, it's that double bond that changes everything. Parts of the carbons at the very bottom are basically going to be, um, you know, not included or will not have a hydrogen atom next to it. I don't really want to get to the chemistry of that, but carbon, if I pull up the periodic table, I'm going to do this really quick for people that are for some reason interested in chemistry for biology. It's kind of the root force of it though. Carbon is 4 away from the noble gases. Carbon wants to become like noble gases, and if it wants to become like noble gases, it will have to gain 4 electrons. That is why you always see carbon in this photo, and let me erase a portion of it. That is why you always see carbon surrounded with 4 different things. Like this carbon right here, this one that I've got a big green dot on, is surrounded with this carbon, this hydrogen, and this hydrogen. It wants to be with 4 things enclosing it. It becomes more stable that way. And that's why you have a saturated fat. But in this case, does it still become, you know, fulfilled with its needs? Yes, you have 2 double bonds, which provides 2 electrons, and then you have 1 here and 1 here, 1 bond and 1 bond. So that's in total 4 bonds. But because you have 4 bonds and one of them is a double bond, you're not going to have it enclosed here, or you're not going to have anything put here because carbon does not want an extra electron anymore. It doesn't want anything else to bond to it. It's happy the way it is, so that's why it creates that kink. And now I'm going to be talking about, you know, where you see a fatty acid and where you see an unsaturated fatty acid. Saturated fatty acids are typically going to be seen in a lot of animals. Humans have a lot of saturated fatty acids. We also have unsaturated ones, but if you kill like a cow, for example, you're going to have a saturated fatty acid. These guys are compact pretty well. Unsaturated fatty acids are not compact because of the kink. Once again, I mentioned that a few times. Note that. And also unsaturated fatty acids are going to be seen in oils because they're not going to be able to be compact. They can't be really a solid. They're going to be more liquidy because of that. They're not, you know, the atoms kind of have more free range with where they're going. So that's that. Compare ionic, polar, covalent, and nonpolar covalent bonds and give examples of each. So an ionic bond, and I guess this is more chemistry and it's going to be a little bit more heavy, is where there is a donation, an ionic bond, ionic, there is a donation of electrons. What do I mean by that? So, if we go over to the periodic table, and be familiar with this is going to be your best friend for this unit. And also in chemistry. But if I look at this and I see sodium, and I then see chlorine, well, all atoms want to be in a neutral or in a more stable state. What does a stable state mean? In a state where they have to have less energy, where they have to do less work. I mean, if I were to, and a great analogy of this would be like, would you rather do a lot of work or do less work and be like more chill? It's, you know, it is what it is. It's the best analogy I can think of on the spot, but would you rather do a lot of work, have a lot, you know, or just kind of reduce your energy, just be in that cool state? You'd probably want to be in that cool state. atoms are very much like that. Everything in the world is, it wants to be in a lower state of energy. So what a sodium atom is thinking is, okay, if I want to be in a lower state of energy, I want to be like neon. I don't want to get into the chemistry of this, so just note that the things on the very far end, these are called noble gases, right? The very far right are the ones that are most stable because their electron shells are filled out. So sodium is like, okay, I only have one electron with me. If I lose my electron, I can become like neon. And if I become like neon, I'm going to become all cool. I'm going to become like chill. Remember, an electron also has some type of energy, a little bit. Okay, so if I lose my electron, I'm going to be nice and dandy. Who can I give my electron to? Who really wants one electron? That would be chlorine, so chlorine can become similar to argon. It won't become argon, but it will become similar to it to stabilize. Okay, so sodium is like, all right, I'm going to give you my electron, chlorine, and chlorine will pick it up and it'll be happy. But when sodium gives off its electron, it becomes positively charged. Why? Because it lost an electron, which is a negatively charged particle, and it has now more protons than electrons. The amount of protons will always be the same to the amount of electrons. So protons, in a normal atom, protons, the number of protons equals the number of electrons. But in an ion, I will always be losing or gaining an electron. So when you form an ionic bond, it is going to be between two charged molecules, all right? And it's going to be either because, or it's going to be because one of them lost an electron, like sodium, to become positively charged, and it's also going to be because someone like fluorine, even chlorine, really any of these on the right, that wants to become to the a noble gas, it'll gain an electron, become negatively charged because there's more electrons now than protons. Um, so then you're going to have these two kind of be attracted, right? Because if I have Na as a positively charged thing, my apologies. Um, if I have Na be a positively charged ion, and I have Cl, or fluorine, Cl, be a negatively charged ion, then these guys are going to become attracted because opposite charges attract one another. So that is going to be an ionic bond right here. So if you have two opposing charges attracting one another, and they have to be negative and positive. They have to be negative and positive. And this is because of a donation of electrons. An example of this is salt, NaCl, that I just created right here. So salt, think of salt. Then you have things called polar covalent and non-polar covalent bonds. Polar covalent bonds, or really what a covalent bond is actually, let me tackle that first. Is the following. Instead of the donation of electrons, you're going to have the sharing of electrons. So if I look at the periodic table, if chlorine wants to bond with oxygen, these guys really can't give one another electrons. They'll be in a weird state. So chlorine will just bond with chlorine, right? And they're just going to be neutrally sharing electrons, right? So when I say sharing electrons, one of the electrons from chlorine will kind of just go over to this guy for a little bit. One of the electrons from the other chlorine will go to this guy for a little bit, and they'll be kind of in this happy state because they kind of filled their electron shells. They kind of got to the noble gases, but they aren't fully like it. They're just kind of in this nice and more favorable state that they once weren't in. Okay? And then you have things that are more specific. You have polar versus non-polar. How do you identify that, and what is polarity? Polarity really occurs due to this concept of electronegativity. So remember how I said that kind of like opposite charges like pull one another? Well, there are going to be more negatively charged and more positively charged, but not fully negatively and not fully positively charged atoms within a bond or within a molecule or a compound. And what I mean by that is basically there are some molecules that can pull electrons closer to them than others can. And what I mean by that is, like, let's take nitrogen, oxygen, and fluorine, for example. These guys, right, they have 9 protons. And that means that if this was to bond with, I don't know, boron, it probably wouldn't. But if this was to bond with boron, which has 5 protons, this molecule right here, right, the fluorine, will actually be able to pull more of the electron closer to itself to become more stable, and boron won't really be able to pull the electrons from fluorine. They'll still pull it, don't get me wrong, but not as well. Fluorine will have a better control over its electrons because, and this is the most important part, it has more protons. More protons means more of a positive charge on the electrons, which aren't as massive and as energetic as the protons are. So the protons will kind of be pulling. And because this has 9 protons, it has a greater charge. It's more electronegative. Okay, more electronegative, meaning that it's really going to be pulling on the atoms, or the, not the atoms, sorry, the electrons that boron has. Does that mean that fluorine is going to be negative? Partially negative. Does that mean boron will be positively charged? No, it'll only be partially positively charged. Okay? But that is a polar bond, that you actually have some form of polarity, some difference. That's what polar means, a difference, right? And that would be a difference in charge thanks to whoever has more protons. I don't really want to get any more depth into the electronegativity concept, but just if you look at a periodic table and you're like, oh shoot, how do I figure out what is the electronegative protons, or sorry, not protons, electronegative atoms? In biology, you're only going to be working with a very few set of atoms, and that's going to be nitrogen, oxygen, fluorine, carbon, phosphorus a little bit, um, hydrogen for sure. But if I were to tell you, hey, how to identify which one's going to be a more um, electronegative atom, it's going to be nitrogen, oxygen, fluorine. Those are the ones that are probably going to be pulling electrons closer to them from the thing that they bonded with. But, and here's a big but, all right? You only get nonpolar things when the electronegativity of the two compounds is relatively the same. So if I have an oxygen double bonding with an oxygen, it'll most likely be nonpolar because oxygen has the same electronegativity. So if there isn't really a big difference in the amount of protons, then you're going to have a more nonpolar substance, and you're going to basically be having a substance in which it isn't really charged. That basically, these guys are really, really nonpolar. There are small exceptions to this, and I think Unit 2 may cover it. You may also have read it in this unit as well. They're called van der Waals interactions. Again, I don't want to get too in-depth with it, but even if you're nonpolar, you will have a little bit of polarity. Nothing too crazy, but there are going to be small moments where an electron may move over a little bit to the right, or a little bit to the right, a little bit to the left, or a little bit to the right, and create some small particle charge for the atom, but nothing too serious in which, like in the case of boron and fluorine will basically like be pulling at 85% of the proton, or of the neutron, not the neutron, the electron of boron closer to it. It won't absorb it, again, by the way. It's not going to take it away. It's not going to the boron's not going to donate the electron. It's going to be sharing it, but it's going to be like an unequal or an unfair share. All right? That's really the chemistry behind what an ionic, polar covalent, and nonpolar covalent bond is. And I guess an example of this is O2 is nonpolar. And a polar compound would be water. And I think that we're going to be talking about the properties of water in quite literally a second. Yeah, I have to draw an example of a hydrogen bond. So let's look at water as another example of a polar substance. Okay, so you have H2O, and it looks like this. Now, like I said before, oxygen, nitrogen, and fluorine are going to be the electronegative substances. They're going to be pulling electrons closer to them. Well, hydrogen only has one electron, so let's just draw one electron as a bigger dot. And it's probably going to be going closer to the oxygen because it's electronegative. It has more protons. It can pull the electrons a little bit better. It has a better grip on them. Doesn't mean that they're going to take them away. Just has a better grip on them, making it a fairly negatively charged particle. Does it mean it's fully negative? No, it is partially negative. But because now the two hydrogen atoms have lost some of their control over their protons, they're going to become more positively charged. Okay, now that they're more positively charged, what does this mean for the interactions of water with water? Like, if I look at a glass of water, why does it look like pretty uniform? Well, and this is the big picture idea here, if I have a lot of this, if I have a lot of these molecules kind of lined up like this, and always draw the charges, it makes things much simpler. If I were to do something like this, well, these guys are going to kind of be a little bit attracted to one another. So you are always going to draw these dotted lines to demonstrate that there is a hydrogen bond. A hydrogen bond is basically when uh, two polar substances, one having to be water, interacts with another polar substance, another substance that is charged through a covalent bond. Okay? So now these guys are kind of connected, kind of closed together. These are called weak interactions. They're not bonds at all. There is no electron sharing, but to stabilize themselves a little bit, to kind of get into that lower energy state, to kind of become more comfortable, the positive and the negative charge kind of attract one another, right? So that is like they become a little bit more concrete. That is a definite, or that is a hydrogen bond. You will have to probably draw that on the AP test. So know hydrogen bonds very well. And I'm going to describe their properties now. Um, why are hydrogen bonds or what are hydrogen bonds? I basically just told you. It's kind of when a water molecule binds to some other not polar molecule, typically water though. Can you identify or draw one? I just did. Is water polar or non-polar? Kind of explained it. Water is definitely polar. Look at it. These are covalent bonds, but there is going to be more of a positive and a negative charge. So water is polar. I'm going to highlight that right there. Know the difference between hydrophobic and hydrophilic. Okay, phobic and philic, what do they mean? And this is going to make your life easier. Philic means love. Philic means love. Phobic means hates. But I'd also just say like not interact. So hydrophilic substances, ooh, I did a voice crack there, not bad. Hydrophilic substances are substances that will interact with water, will kind of come closer to water, okay? And those are always going to be polar substances because they're charged. Only charged molecules are going to interact with other charged molecules, okay? So these are always polar substances. Hydrophobic particles are going to be non-polar. They don't really have a charge. They kind of bump into water. They're like, oh well, what am I going to do with you? Nothing, right? They're going to go away from water. Non-polar substances are going to be more interested with non-polar substances, while polar substances are going to be way more hydrophilic, meaning they want to interact with water, okay? So I'm going to write this down here. Hydrophilic are always polar, charged substances that want to interact with water. Hydrophobic is another word for really just non-polar. These are non-charged substances, or weakly charged, nothing too crazy. And these properties of hydrophobic and hydrophilic will really just determine how water interacts with things. So now we're going to get to adhesion, cohesion, and surface tension. So adhesion, I believe, is very similar to cohesion, okay? But there is basically one distinct difference. They both are derived from the fact that it's water dealing with something, but it's what water deals with, okay? Cohesion is when water deals with water. So when water, water hydrogen bonds with water, and I'm drawing my charges, so it hydrogen bonds, remember the dashed lines again, with water. Boom. But adhesion is when water interacts with hydrophilic substances, things that are not water but are also negatively or positively charged covalently, right? So polar substances. So this is for polar substance with water, and this is just water with water. Okay? So for example, we have sugars, right? Sugars are fairly polar substances because they are made of CHO. These two substances, or these guys right here, are going to be the fact that it makes it polar. But in your cell, and I think this might be a good thing to remember because I've seen it on the AP test a few times, the cell is made of this thing called cellulose. Especially in plant cells. It's what the plant walls are made out of. Cellulose. It's a hydrophilic substance. And let's say plant wants to get water all the way up into its leaves. right? Well, it has to go against the force of gravity because gravity's pushing down on water. How, the question is, how does water kind of get up there? Well, water gets up there because water will bind to kind of like the sides of these plant wall cells and kind of get dragged up. Like it's going to be, ooh, I'm going to interact here now. I want to interact here now. Boom, boom, boom. And of course, there's a lot of water on the ground, so it's kind of pushing it upwards. And that's the adhesion. The reason the water molecules kind of all compactly go up is because, sorry, that's cohesion. The reason all the water molecules all go up together is because that's cohesion. The water molecules are attracted to the water molecules, right? But at the same time, they're able to kind of climb the plant wall cell because of adhesion, right? The plant wall cell is a hydrophilic substance. It is made up of oxygen, which is a electronegative substance, which is making the entire thing a polar substance, which makes it attractive to water. Okay? Now, use these ideas to understand surface tension. So, when I have a body of water, think of a bunch of molecules, right? This is H, 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 O, H, O, H, H. There are a lot of molecules in the air, okay? And these could be charged negatively, okay, positively. And they're kind of pulling on the water that's kind of at the top of the surface to kind of pull it up. But at the same time, and of course, water doesn't do that because of surface tension. As water is getting pulled upwards a little bit, at the same time, because of adhesion, all right, sorry, because of cohesion, apologies, because of cohesion, these molecules are kind of getting strained, right? So this guy is going to be bonding to this guy, and it's going to kind of create this like barrier, this umbrella, this pressure around, right? Because there's a pull going like upwards, right, from the gas molecules in the air, but then the water molecules are pulling it downwards, right? Because remember, there's like, imagine this like umbrella as one group of water molecules at the very surface. Then there's water molecules all on the bottom, and they're pulling down on the water. Each of these water molecules are kind of pulling onto one another, right? Like imagine this bond right here. They're kind of pulling to the left and right. Then there's going to be water molecules on the bottom here. And this is going to kind of be pulling, these are intermolecular forces, they're going to be pulling the water down, so you kind of get this umbrella on the water, and because of this, you actually see like things like geckos, or I don't even know the animal, but the animals walking on water because of their really cool, like fast web feet. They're kind of able to utilize this surface tension. All right, so that is surface tension. The bond that forms between a sodium atom, and this is number 20, and a chlorine atom is considered a blank bond. Well, let's look at sodium and let's look at a chlorine molecule. Sodium is right here. Oh, okay, and chlorine is right here. Interesting. Okay, so if sodium gives off one electron to chlorine, it can become very neutral in the sense that sodium will become like neon, chlorine will become like argon. It's ionic. Another way to identify ionic bonds, and this is a pretty good thing, identify ionic bonds besides the fact that they donate electrons, right? An electron is going to one end or losing. Identify ionic is really this. If it's a salt, it's going to always be ionic. And this is the big point, if it's a metal plus a non-metal, they will almost most definitely be ionic bonds. And I want you to look at this. All the metals on the periodic table are going to be on the left of this ziggity-zag line where all the yellows are, okay? So on this zigzag line right here, I don't know why I said ziggity-zag, that's kind of weird. On this zigzag line, God, I'm tired. On this zigzag line, everything to the left of it is a metal. So if it reacts with something to the right of this zigzag line, so anything here, these blue and red stuff, if it reacts with it, if you see like these two elements connected with one another, it will 99% of the time, at least for this class, probably be an ionic bond. Okay, that's just like a pro tip kind of. So it's going to be an ionic bond. I don't know why I did that. Boom. Describe the properties of monomers and the types of bond that connect the monomers in biological macromolecules. Okay, what is a monomer? It is just a subunit of a polymer or just one block. So think of like a chain. If I were to draw a chain of like, I don't know, cubes, a monomer would just be one of it. a polymer, and I'm just going to kind of go beyond what the question is asking because you're going to get this in subsequent questions. A polymer is that entire chain. It is everything combined of those monomers, okay? And a macromolecule is just a polymer that is composed of monomers, all right? That is what a macromolecule is. So, there are 4 big picture macromolecules you have to know on the AP test. You have lipids, you have nucleic acid, you have, I don't know if I spelled nucleic acid right. Forgive me if I didn't. And I guess you can call them fats as well for lipids. Then you're going to have carbohydrates, and finally proteins. So these are the 4 big macromolecules, okay? These are the polymers. These are the polymers that really your body in every living system uses. So, what are the monomers of each of these? Lipids is a fatty acid. Nucleic acid is a nucleotide. So basically when I say that the monomer of a nucleic acid is a nucleotide, that means that if I get this long chain of nucleic acids, or if I get a nucleic acid, it is going to be this long chain, and there are going to be small units that are always continuously repeated. It is the same unit, okay? And that will be a nucleotide. Same for carbohydrates. If I get a long chain, which will be a carbohydrate, and I look at one of those subunits, it will be the same subunit as the one before it, and it will be just that monomer of it, which is I believe it's a monosaccharide. That is the correct term. And then you have proteins, the same thing. An amino acid is going to be its subunit, but I'll continue to talk about that basically later on. Unless I can. I'm looking at these questions. I think I have to kind of talk about them. All right, yeah. Actually, let me talk about it real quick. So, polymers and the respected monomers. So, if I were to get a lipid, I'm going to be seeing a fatty acid chain, or a fatty acid kind of. If I were, yeah, I guess that's fair. Lipids are the exception to monomers, by the way, or sorry, polymers, because they come in different ways. There's phospholipids, which you'll learn about in Unit 2 in more depth. Then if I get nucleic acid, this is the stuff that transmits DNA, by the way, I will have a nucleotide as its monomer. If I have a polysaccharide, is the scientific term. I don't know how to spell it, aka carbs. You're gonna have a monosaccharide, and then finally proteins are amino acids. Okay, and when I say amino acids, if I get a protein, every amino acid should be relatively the same in structure, and it's gonna be just chained together. Okay, now I'm gonna pull up every single one of these. A protein structure. Primary level. Actually, yeah, here's a perfectly good example. If I were to look at this protein, okay, this protein has .1, .2, .3, .3, .4, .5, .6, .7, .8, okay? Or even here. These little dots are the amino acids, okay? These are the monomers of the protein, or the polymer, okay? And proteins are connected with peptide bonds. Now, if I were to show you a polysaccharide, on the other hand, you see this little bench right here, this O? That is called a glycosidic linkage. For nucleotides, or nucleic acid, you see these backbones right here? Those are called phosphodiester linkages. They're called linkages because they connect things. And if I were to finally show you a fatty acid structure, the things that connect the monomers, or a lipid. Sorry, a lipid. Damn. There we go. A lipid, you see this H thing right here, this weird stick at the side for both of these on the left far side? That is called glycerol. And a lipid is made of glycerol and a fatty acid chain. Okay? This bond right here, you see this O? These O's right here? Those are what connects the glycerol, which is a type of alcohol. So think of it as plus glycerol. That is also the monomer of a lipid. it is with an ester linkage. The glycerol and the fatty acid are connected through this thing called an ester linkage. Okay? So, how do all these things kind of connect? Like, how do I get one nucleic acid, one subunit, and basically say, hey, make a bond, right? Because they're all kind of floating everywhere, right? In your cell, in a body, they're all everywhere. So what do I got to do to kind of make them get closer? Well, assuming that these two things kind of collide, right, with one another in the body or in the cell, right? It's going to be in the cell most likely. They're going to go through this thing called a dehydration reaction. A dehydration reaction. That is something that you need to know, because when a dehydration reaction occurs, all right, you will for sure be using water in it. So think of hydration as water. You're not using it, but rather you are creating it. If I want to connect these two things together, I will connect them, but I will also create water as a result. It's a fundamental guarantee. And as a result of this, this ends up actually consuming a lot of energy. All right? Because I want to make something. If I want to build something together, I use a lot of energy. Now, the converse happens when, let's say, I want to break down sugar, right? I mean, think about, you ever hear of, like, ATP synthesis? Maybe that's too far. Let's say when you want to break down food, right? You're breaking down food, you're breaking down food, you're breaking down food, right? Let's say some sugar. Well, sugar, like we said, is a carbohydrate, and a carbohydrate, right, is a polymer. So it's going to be made of these things right here. And if I want to cut these bonds away, I will bring in water, right? Assume this is H2O, and water will kind of get in between these two bonds and separate them. So then I'll have, one guy here, and then the rest kind of here, and like, I'll add like a H from the hydrogen here, and I'll add like an HO here, and that kind of like stabilizes these two guys. It's like, hey, I know you lost a little bit, but let me, put you guys together. So dehydration reactions use energy, and they are for combining monomers to make polymers. Hydrolysis, which I just described, is, and dehydration is also make water as a net result. Hydrolysis, on the other hand, and by the way, you can kind of count how many times you make a molecule. Sorry for sidestepping again. You can count how many times you or how many molecules are within a thing if you count the amount of water molecules that are made. Oh, if I have 2 water molecules, that means 2 bonds were formed. So, that'd probably be like 3 polymers in the entire thing, because polymer A and B kind of mix together, and once those two things are together, they'll mix with C, make ABC. All right? So, you could think of it like that. Hydrolysis, on the other hand, is using water to break apart a polymer, all right, into monomers. This typically releases a lot of energy, right? If I want to break stuff down and get into a smaller thing, I have to release energy to go down. Like, each time I go down, I lose energy. So, yeah, that is how monomers and polymers are formed in the quickest way possible to describe. Be able to recognize a structural diagram of monomer and various organic compounds. The best way I can tell you is for lipids, it's always going to be this fatty acid chain that you're going to have to recognize. There are exceptions. There's a thing called a phospholipid. Phospholipids have this phosphate group attached to it, but it will still have the stupid fatty acid chain. But it's only going to have 2 of them instead of 3 sometimes that you see. Then you're going to have proteins, all right? And I'm going to just do the primary structure of a protein. And you will notice, actually, hold on, an amino acid. All right, if you want to figure out what a protein looks like, look at this. Look at this. This is called an amino acid. That's one of the polymers that you have to know. That is correlated to a protein. If I have carbohydrates, they will always look like in two ways. Either this weird hexagonal shape, or either a hexagon or a pentagon. All right. These two right here, 1, 2, 3, 4, 5, 1, 2, 3, 4. These are going to be what carbohydrates look like, or this. Another way you can tell, and I think you should write this one down in particular because I've seen it a lot on tests, is what specific polymers are made of. This is a really good thing to remember for your test. Carbs are made of pentose or here pentagrams. They look like pentagrams. Pentagrams? No, pentagons. Oh my god. Ignore that. That's embarrassing. Pentagons or hexagons. And they're only going to have the following three elements in them: carbon, hydrogen, and oxygen. Okay? So if you're dealing with something and you don't know what it is, if it only has carbon, hydrogen, and oxygen and like that, you will most likely have a carb. Then you have lipids. Lipids, and also remember like the if you get the word glycosidic linkage, right? That's what you kind of really have to remember for that. Lipids, you're going to have a fatty acid chain, so they're just going to be really long. Like you can tell if it's a lipid. And they're also going to contain only carbon, hydrogen, and oxygen. But the catch here is that you could tell it's a chain. You have to look for the chain. You have to look for a chain again, for a structure. And they could also contain phosphate sometime, or phosphorus, because of a phospholipid. So it could be a lipid if you have like, it's like, hey, you have all these elements and you include it into a cell. These were the things that were be able to be made. If you only get carbon, hydrogen, and oxygen, you're probably going to be like, oh, only carbon and lipids can be made. Now I have proteins. Proteins, if we go back and look at them right now, are made of amino acids. Okay, so if I were to look at amino acids, I want a really good photo of. You have this carbon right here. All right, here, this is perfect. This is a perfect diagram for an amino acid. You have a carbon, so an amino acid. You're going to have a carbon. You're going to have an oxygen. You're going to have a hydrogen. And finally, you're going to have a nitrogen. That is the most important part. If it has nitrogen, okay? Now, if we look at a amino acid, and let's look at this one right here, you want to know and identify these really well, because you're going to have to work with proteins later on in the unit, and also in other subsequent units. Know that there is this part called an amine group, and an amine is just NH2, okay? Know that chemical formula. Then you have a carboxyl group at the other end, COOH, okay? Know what that is as well, okay? That is carboxyl. Now, the reason these are two significant things is because when a protein wants to be made, these actually kind of interact with one another, and they end up forming some type of weird bond on top of one another and creating this like weird structure. You have this thing called an R group, which are just chemical. R groups are, the best way to explain it is they're actual chemical things that are attached to it. It's not the letter R naturally. It's just that because amino acids vary, you're going to have different things or different chemicals for the R group. But the R groups are very important, and I'll get into how that's important for a protein just in a second. So there's that. And then if you want a nucleic acid, know that they're made of nucleotides, and nucleotides are made of the following: carbon, hydrogen, oxygen. And if I were to pull up a nucleotide, the best way, okay. So a sugar, right? It's made up of three things: a nitrogenous base, a sugar, and a phosphate group. Sugar, like we had already said, is a carbohydrate. Carbohydrates are made of CHO, so that's where you get your CHO from. You have this nitrogenous base, which the wording of it is just N. And then you have a phosphate group. That is the P. That is really going to distinguish a protein or an amino acid from a nucleic acid if you only are given elements. That is how you're going to identify. CHOMP, okay? Know those acronyms: CHO, CHO, COHN, or CHONP, okay? These are what you're going to be able to kind of distinguish. Oh, I know that I'm going to be growing nucleic acids if I put a lot of phosphorus into this thing, or that's what the cell would be able to make, all right, if I were to give it these ingredients, kind of. Know kind of what the structure of nucleic acid is also. This is a better diagram. The nitrogenous bases are the most important part you have to know, because these are where like the DNA is actually made. So if you were to look at this photo right here, these A, T's, G's, and C's, these are all the nitrogenous bases, and they hydrogen bond, all right, they hydrogen bond with one another to kind of form your double helix that you always see with like DNA, like this structure right here, all right. The phosphate groups are always on the backbone. at the very end, and so is the sugar. Okay, so that's that. I know I'm kind of rushing, but kind of want to get this done with for you guys. Be able to recognize the differences. We did that. Be able to recognize the structural diagram of a monomer of various organic compounds. You just got to stare at organic compounds for those. But I've kind of gone over each one of them. I'll have them up again. Nucleotide, amino acids, um, fatty acid plus glycerol, and lastly carbohydrate structure. Right? So know what those are. Know what those are. Know what these are. Know what this looks like. Alright, that's what it's basically saying. In amino acids, what is meant by the R groups? What is the carboxyl and amine group? I kind of already described it. One more time, though. If I were to show you an amino acid, the COOH is the carboxyl group. The NH2 is the amino group, okay? These are significant for this part of the... So, a protein... Actually, let's see if that's part of your questions. It's not, but... I sure as hell hope so. It is on the. You're gonna get this on your test, basically, is what I'm trying to say. A protein is made up of four structures. A protein structure, okay? If we were to look at this, and I'm gonna go over it real quick. Okay, your DNA. Actually, I don't even think you have to know about it, but your DNA, the nucleic acids, determine, and just memorize this. You'll understand what I mean by Unit 4. Your nucleic acids are going to be determining the primary structure of a protein. What this means basically is, hey, look, like I said, amino acids come in different ways because the R groups can change. Like, look, these are all amino acids. They all have the NH2. They all have the COOH. They all have that, right? But they all have these different, like, these weird red things, right? Think of these red things as the R groups. They are the R groups. So each amino acid comes in as a different thing. And because each of them is different, that is what's going to impact the primary structure. So the actual amino acid. So when they all bond... Let's call this amino acid A, this one A, and this one B. All right, just the sequence of amino acids. The sequence of amino acids is the primary structure. Now, what happens in this primary structure is that as I keep on making more and more of these, the NH2s and the COOHs kind of begin to fold on one another. So I will create this like thing right here. They will begin to fold on each other. And as they begin to fold on each other, they'll kind of form bonds. These hydrogen bonds make something called an alpha helix or a beta helix. Alpha helix or beta helix. These are the secondary structure. Okay? Beta sheets and alpha helix sheets, these weird things. And they're held together. So secondary structure is the alpha / beta helix sheets. They're held together by hydrogen bonds. Okay, because like the COOHs and the NH2s, remember nitrogen was an electronegative atom. COOH has oxygen, that's going to make it electronegative. They're going to have different like polar charges, and those polar charges will then typically interact with one another because they're slightly negative, slightly positive at different ends, so on and so forth. You're going to have that as a secondary thing. Next up, you're going to have the last stage. Sometimes it is called the tertiary structure, I believe. No. That is not the tertiary structure. Structures of proteins. I don't know why I'm going. This is so unprofessional. Oh yeah, tertiary and then quaternary. Yeah, no, I'm right. My bad. This is really determined by the R groups. So know that this is determined by R groups, and there are many types of there are many ways that the R groups can bond with one another. So R groups in proteins, if you were to look at this, each of these that are highlighted are the R groups. The rest of the protein or the rest of the amino acid is going to be very much the same. But if you want to get the real structure of the protein, okay, the real 3D shape of the protein, the R groups are what determine it. R groups, tertiary structure. Remember that. And it's basically through either polar bonds from the R groups, right? So if I were to look at this CH2, oh, I have an NH2 right here. That right there, that nitrogen is going to be electronegative, and I know that if this nitrogen is electronegative and I have this glutamine right here, these two can actually bond because then it has another NH2 here. The hydrogens are going to become positively charged because it's a polar covalent bond. The nitrogen that's negatively charged here and the hydrogens that are positively charged here will kind of bond with one another and bring these two things closer together. Okay, they will bring the two amino acids closer together, creating a more 3D shape. You have these things called disulfide bridges as well, that make part of the tertiary structure. Disulfide bridge. You have ionic interactions. Ionic interactions. And you have hydrophobic interactions, so nonpolar interactions. Hydrophobic interactions are very important. Know that for your test. Typically, they're going to be on the inside of a protein. So if I were to have a protein, you know, dot, 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 going all the way around here, this part that's kind of like caved in right here is going to be very important, okay? You're probably going to have like nonpolar stuff here because they're away from water, okay? Um, they don't like the water. They don't really want to interact with it. They're going to be kind of caved in. They're going to be sheltered, okay? So that's where you can identify nonpolar or hydrophobic interactions. But these are the things that make up the tertiary structures. A quaternary structure is like the last stage, and that's when another protein kind of wants to bind with another protein. So if these two kind of want to bind together, that's also with the R groups. Similar bonds, by the way, polar bonds, disulfide bridges, ionic interactions, hydrophobic interactions. Those are all the things that are going to kind of be connecting these guys together. Okay, so note that primary structure, just the sequence of amino acids. These are determined by the nucleic acid. Boom. Next one is going to be your secondary interactions, and those are by the alpha helixes and by the beta helixes. And lastly, you have the tertiary and quaternary structures that are determined by R groups. So just know that. Know what type of bonds have each one of them. Peptide bonds are what control the primary structure, right, because they're between the amino acids. The secondary structure is hydrogen bonding. And tertiary structure is going to be between the R groups, and these are the type of bonds that you're going to be really seeing within them. So that's that. I covered that. What is the relationship between monosaccharides and polysaccharides? I kind of determined that. Monosaccharides are like the monomer of a polysaccharide. Boom. That's that. What are functions of carbohydrates? Carbohydrates can really vary. You have things like cellulose, which kind of develop plant walls, like I talked about how adhesion works. Then you also have carbohydrates used for a burst of energy. I forgot the word glycogen. Glycogen in human bodies is very similar to cellulose in structure. There's a slight difference, but glycogen in us is used for energy because if I want to go dart really quickly, my body has to break down some food for me to kind of have the energy to move around. It'll use sugar very quickly. I think you use about all the sugar in your body within a day. Once that's gone, you're going to start using fats. What are organic compounds? What organic compounds use glycerol and fatty acids in their building blocks? Like I told you, know what the monomers are. These are monomers, so that's going to be a lipid. I'll mark that here. Which of the following is a function of the nucleic acids? Nucleic acids are part of your DNA, so they are going to be containing and transmitting genetic information. Okay. In general, how do organisms grow, reproduce, and maintain organization? This is really just through utilizing energy for themselves. They break down energy to kind of grow and live. There's mitosis, stuff like that. Not really a great question, in my opinion. Just kind of know like the stuff I've listed above. That's kind of what like the organisms use to grow. Understanding that a change in the subunits of a polymer may lead to a change in the structure or function of a macromolecule. That is absolutely true. If I were to have a molecule or a polymer of 5 units, all right, and I were to tell it to kind of be like, okay, I want you to interact with this circle right here. Well, the circle is going to recognize these 5 units, but if I were to kind of have a hydrolysis reaction breaking off one of these units right here, and I only have these 4, it won't be able to bond as well. And the best way I can show you it is through a protein. I want to show you a protein structure like this, okay? This looks weird to you. It looks weird to me. I don't even understand it perfectly right now. I only finished AP Bio. But if I were to look at this protein right here, I see all these weird squiggly lines, and I should know that these squiggly lines, these are, by the way, this is the tertiary structure of a protein, that these squiggly lines will interact with another thing that will recognize those squiggly lines because it'll only work for those squiggly lines, okay? Like, if I were to have that, as a kid, right, if you had that box where it has like that little square, that circle, and that triangle, and you have to put the actual triangular block into it, right? Or if I have to put that circular block into it, it'll only recognize that circle. So if I were to slightly change that shape of the circle, kind of like cave it in a little bit, this circle isn't going to really work anymore to recognize that. So in a way, it'll actually change, well, the structural change or the change in the subunits, because when I mean change in the subunits, like if I were to change the amino acids, for example, I create a totally different primary structure. That primary structure may change the way the bonds work, so the function of the macromolecule may be hindered. When two organic monomers are joined together, a molecule of water is formed. This is known as a dehydration reaction. I kind of already showed this. Dehydration reaction. What is the opposite process called? When does it occur? Hydrolysis. You gotta just bring water to a polymer. I already discussed it. By the way, these are gonna react. These are gonna need things called catalysts. You'll learn about it in Unit 3. But yeah, a friend tells you he was trying to cut all possible sources of fats out of his diet. Using your knowledge of the roles of lipids in your body, what would be the most effective way to convince him this plan isn't very healthy? Fats are your biggest energy source. And if you basically just say, screw it, I'm not going to, eat any fat anymore, that's going to be kind of dangerous. You're going to be running really low on energy. So, and the reason for that, by the way, like why fats are really good for energy is like, look at this. There is so much carbon-hydrogen bonds all throughout here, even in an unsaturated fat. But if I were to look at a carbohydrate, look how small this is in relative term, right? I mean, sure, they're going to be bonded together between glycosidic linkages, but like, you're going to have millions of fatty acids and millions of sugars, but with millions of sugars, you're only going to have these like 4 or 5 blocks. With fatty acids, you're going to have millions of these carbohydrogen chains. They're called hydrocarbons in chemistry. And that's just a lot of energy. And that's really what your body's going to use to kind of create its energy if I want to break it down, right? So I would tell my friend, no, you're being a total idiot. How about you cut down, maybe sugar? How about you cut down on, I don't know, bad drinks? Stuff like that. So there's that. Review the structural difference between RNA and DNA. For each item in the following list, place an R if it applies to RNA and D if it applies to DNA. So what I'm actually going to do instead for this last portion of the video is kind of talk about what RNA and DNA do. So DNA, okay, is basically where all your genetic information are stored, and that is kind of the double helix, my best interpretation of it. And you also have, and this is DNA, and it is only for genetic information storage. All right, this basically tells me why I look the way I look, how I work. Okay, that is DNA. Now, if I want to genetic information storage, if I want to express that DNA, make that DNA into basically like, these are the command. This is the command center. This is the best way to say it. It's the command center, or the command control, the command panel. If the command panel wants to say, hey, I want you to make this protein, I'm going to make RNA. Okay? Now, what is DNA in its actual word? Deoxyribonucleic acid. What is RNA? Ribonucleic acid. Okay? Really, the difference is minimal, but it's still significant enough to differentiate these two. You're going to learn more about RNA later on. It's really, really huge. But RNA is single-stranded. All right? That is a big, big difference when you're looking at DNA and RNA. RNA is going to be single-stranded for the most part. There are times when it kinks around, but don't worry about that. Note that it's single-stranded. So I'm going to make a little chart here. DNA versus RNA. DNA is going to store genetic information. RNA is going to be helping express genetic information. You'll learn about that in a later unit. I will go over that in a video. Then you have DNA is double-stranded in a double helix way. RNA is single-stranded. Okay? Another thing that is very important to note, and I think that on this test you may have to understand this pretty damn well, is the difference in the sugars. Deoxyribonucleic acid versus ribonucleic acid. If I were to look at them, there are two distinct differences. Here, the sugars. Now, this is going to be a very nuanced thing, okay? But the sugars are going to play a huge role. This H right here and this OH right here, they look almost exactly the same. But if you go to the right side of these molecules, I see this H right here, and I see this OH right here. Ribose sugar, all right, has the other OH. Deoxyribose only has one hydrogen atom. It lacks that oxygen atom. Know that damn well, because I'm quite certain on the AP test or with your bio teacher, they may throw around some really, really hard question to differentiate the two, because you may not know what, the nitrogenous bases look like. You only may know, like, have a photo of the sugar, and you would have to be like, oh, this one has an oxygen, this one doesn't. So again, DNA is only one OH, so the hydrogen is missing an oxygen. The ribose is going to have two OHs. So I'm going to label it right here. This is DNA. This is RNA. And really look at this right here. This sugar on the right. All right? On the right of the sugar, so on the, the pentose, this is what's different. And there is one more thing, and this is going to be a little bit more significant, that is different, but I just wanted to point this one out because it's pretty valid. Is deoxyribonucleic acid versus acidic ribonucleic acid. Perfect. Here. Remember how I said that there are nitrogenous bases within a nucleic acid? There's the phosphate group, which is the only part or only macromolecule that contains a phosphorus atom because of this, besides a phospholipid, right? There's that phosphate group. Then you have the sugar, right? The sugar is what also differentiates RNA versus DNA. And lastly, and most importantly, is RNA has a replacement of the four nitrogenous bases that are used for DNA. One replacement. DNA has cytosine, guanine, adenine, thymine. I always think of AT, GC, because that's how they base pair. A always goes with T. G always goes with C. Okay? And if you were to look at it, look at this T right here. I'm going to, zoom in on this a little bit. If you were to kind of see this, look at the red right here, and look at the yellow right here. Red, yellow. Red, yellow. It's always going to work like that. It will always complementary base pair with an A and a T. Same for G and C. Okay? Now, again, you can also see that these two are structurally different. One is single-stranded, one is double-stranded. But the nitrogenous bases differ because RNA has this thing called uracil. It is a replacement of thymine. Okay? Uracil is this orange thing right here, and it is going to replace thymine. All right? So that's really it. I want everyone to note that most RNA is not only found in humans, but also viruses. Viruses are another good example of something that contains a lot of RNA. But yeah, let's go now with this entire review. What is composed of nucleotides? R and D. Regardless of what this is, these are all nucleotides. Like, this is still the phospho diester linkages all in the back, right? You have the sugar and phosphates, as it says right here. They both have, sugar and phosphate and nitrogenous bases. It's just that the nitrogenous bases vary a little bit, and the sugars vary a little bit. But it's still like the same basic structural components, just maybe with a few atoms here and there removed, okay? And sometimes in a nitrogenous base removed in the case of RNA. Contains the sugar ribose, right? Remember that extra OH, that is with ribose. Is the hereditary material of all cells, that is DNA, right? DNA has the genetic information. Contains a phosphate group, both of them, right? Phosphate groups are composed of the nucleotide. They're that backbone. Sometimes contains the nitrogenous base adenine. That's both of them. Sometimes contains the nitrogenous base uracil. Only RNA. Why? Because uracil is the replacement for thymine, which is in DNA, and that's the answer to G. Is double-stranded. DNA. Is single-stranded. RNA. Okay. With that being said, I think that's mostly it. I'm surprised there is nothing about denaturation, but I want to kind of touch on denaturation a little bit because I think it's pretty important for this unit as well, or denaturation. This is the last topic. Denaturation is basically when you misfold a protein. This only occurs if you raise temperatures, okay? Or change pH, or solution. What do I mean by that? Well, um, temperature. Bonds break when you add energy. Temperature is a form of energy. So if you think of a protein, right? This is the primary structure of a protein, right? And it has hydrogen bonds connecting it. Remember, these are very weak interactions. If I just increase the temperature a decent amount, not very small, but a decent amount, those hydrogen bonds are going to be the first thing to break. And when those break, you can imagine that protein kind of like, swinging around now. It's like way less compact. You're going to only have like the bonds between these two things. Okay? You're not going to have, you see how it's like really, really compact here? You're going to have like a much, much more open space. And if it needs to bind with something that only recognizes that really compact structure, it's going to be super confused if it's trying to bind with this like really loose structure. So temperature destroys it. pH also destroys it. Why? Remember, pH is a concentration of H plus ions. Or protons. If there are a lot of protons, they're going to go try to bond with the polar R groups, right? So if I were to draw this, so it's going to impact the tertiary structure of things. This is also Unit 4 material, but it's going to be maybe covered in Unit 1. So if I were to put a lot of H plus ions here, imagine the R groups are kind of bonding together at first together. Well, if I put an H plus ion instead, they may go bind with other parts of the R group and slightly changing it. You're just basically adding stuff that doesn't need to be there, and that will change the structure of the protein. All right, and if it changes its structure, it's going to lose its functionality. Okay. Lastly, solution. Remember how I said proteins, like if I draw this squiggly line, have this like encoded portion that's like typically hydrophobic because it's in kind of caved in away from like water where most proteins are going to be in, right? Think of these dashed lines as water. Well, if I were to remove this protein from water and put it into a hydrophobic substance, right, into this like let's say glass beaker that is a hydrophobic substance, well, these hydrophobic material are going to be attracted to it, and these hydrophilic material are going to be unattracted to it. They're going to repel one another. So then the actual shape of the protein is going to go inward, or kind of fold inside out. Okay? So now you're going to have like some weird thing, and it loses its function because it has denatured. When you denature something, all right, it loses its function. The structure gets altered. It cannot do its job. Okay? So denaturation only occurs with temperature change, increase or decrease, a change in pH, whether it be increase or decrease, or changing the solution, typically from a hydrophilic solution to a hydrophobic solution. All right? With that being said now, I wish you the best of luck on your test. You should really try out question 35 to talk about, the way proteins work. And I will make a video about statistics, right after. Good luck.